nanoHUB-U, online instruction.
Welcome back. I'm Professor Rickus. In this lecture, we'll have an introduction to feed-forward loops, an important motif in biological networks. So we'll introduce the feed-forward loops, and we'll look at some networks in which they are found to be particularly enriched, both E. coli and yeast transcription factor networks, as well as many neuronal networks. And then we'll take a look at some subtypes of the feed-forward loop, including coherent and incoherent, as well as the integration logic and an OR. So take a few minutes on your own and consider a subgraph of 3 nodes, in this case for us, genes. So each dot represents a node or a gene. And think about how many possible ways can these 3 genes be connected. So connect them by drawing arrows. Include direction in your connections, so you have directional edges. So a connection could go this way and this way. But don't worry about sign as to whether it's activation or repression. Just that one node has an effect on the other. Now, how many unique ways can you connect these 3 genes? I'll give you a minute to do that, so go ahead and pause. Okay, now that we're back, hopefully you came up with something close to these 13 possible connection patterns between these 3 nodes. Again, we're ignoring sign, activation, or repression, but we are looking at direction. And if we look at these 13 different possible unique connections, number 5 here is our feed-forward loop. And in our feed-forward loop, if we sort of rotate this and now assign some names to our nodes, so if we consider gene X, gene Y, and gene Z, we can see that X can have a direct effect on Z, or it can have an indirect on Z via Y. And this is our feed-forward loop. So just to sort of compare to a loop that you may be more familiar with, you may have spent a lot of time thinking about feedback loops. So in comparison, the feed-forward loop versus the feedback loop, you can distinguish these again by this directionality of these indirect and direct paths. As we saw in the last slide, where X can have a direct effect on Z or an indirect effect on Z via Y, in our feedback loop, our effect of X on Z now feeds back via Y. So the direction of that indirect loop, whether it's going from Z to X, is our feedback loop, whereas when it's going from X to Z in both directions is our feed-forward loop. So within our feed-forward loop motif, there are actually 8 possible feed-forward loops when you consider sign now, activation and repression. And look at all the possible combinations of activation and repression. And we can now divide these 8 subtypes into 4 that are coherent feed-forward loops and 4 that are incoherent feed-forward loops. What coherent means is that the effect of the direct and indirect paths have the same sign. So let's look at our first coherent type 1. So if X has an activation or positive effect on Z, and X activates Y, which activates Z, both paths, the indirect and the direct, are going to have the same effect of activation. Through either path, X activates Z. And we can look at the effect of the indirect path by multiplying the signs within that path to determine this total sign effect. So positive times positive, activating an activator results in positive activation. Whereas look down here at our type 1 incoherent feed-forward loop now. So X again has an activating effect on Z, and X again activates Y, but now Y is a repressor of Z. So we are now activating a repressor. The net result of that is repression. Positive times minus equals a negative sign. So the effect of the indirect path in our incoherent type 1 now, or of our indirect path, is repression or negative. Activating a repressor is repression. Okay? So the next question is, how does Z, at that Z gene, how are the inputs from the direct and indirect path integrated? And this is where our and or integration comes into play. And so let's take a look at this mechanistically of how we could, at the level of gene, get and integration. So in the case of and integration, both our active activator and our active repressor and our active transcription factor, they could both either be activators or repressors, but both must be bound at the regulatory sequence to have the effect. Okay? Whereas in OR integration, either X or Y need to be bound at the regulatory sequence. So either X or Y. And so this is one way in which we can get and or integration at the Z gene. So feed-forward loops are enriched in E. coli transcription networks. Here is a partial transcription network built from databases of protein transcription factor network interactions in E. coli. And here now, the blue nodes are highlighted in blue in order to indicate their participation in feed-forward loops. So again, here's our feed-forward loop in our blue, and we can see now all of these feed-forward loops that can be found within this highly connected network. And so what do we mean by they are enriched in our network? So one way to quantitatively test this, in other words, to say, in a network that we're looking at, are there more feed-forward loops than you would predict if the nodes were just randomly connected? And so that can be tested by generating random networks. So for example, say we look at an E. coli network that has 424 nodes and 519 edges. So that's the real network. And within that network that was looked at was found to have 40 feed-forward loops in that network. So you can ask the question, how many feed-forward loops would we expect to pop up in our network if those nodes, those genes, were just randomly connected? So we can do that, right? So we could take 424 nodes and just keep adding edges at random, connecting them, until we sum up to the same number of edges, in this case 519. And we can do that multiple times and ask, on average, and with what variation do we observe feed-forward loops in those random networks? And then we can compare. So in this case, they did that, created random networks of the same size in terms of nodes and edges as this defined E. coli, real E. coli transcription factor network, and found that on average in a random network, you would only expect about 7 feed-forward loops if you just plus or minus 3. And so now you can do the statistics and calculate the Z-score and say, are your feed-forward loops enriched in your real network? 40 is clearly bigger than 7 and shows that statistically there's a statistical difference in our real network. So we can say that feed-forward loops are enriched in that real network compared to random. This was also done for a yeast transcription factor network, again seeing a similar degree of enrichment in that system. So what about our subtypes? So they also went in and asked the question, of our 8 subtypes of our feed-forward loops, are some of those enriched more than others? And it turns out the answer was yes, that coherent type 1 and incoherent type 1 feed-forward loops were highly enriched in both the yeast and E. coli transcription factor networks that they looked at based on existing databases of connections. And so now this also raises questions about what design features do these motifs offer? If this biological network has evolved over time to be enriched in these feed-forward loops and is maintaining that enrichment and accumulated feed-forward loops over time, what advantage or what features do those feed-forward loops impart that are advantageous to the cell? But let's first also look at some other networks. So it's not just in transcription networks in which we see feed-forward loops. So another really interesting biological network that's definable exists in the neuronal network of the model system C. elegans. Now, C. elegans is a nematode, and it's a really neat model system for neuroscience as well as biological networks because it has a definable network of neurons. In other words, it has a tractable and fairly consistent wiring diagram of its nervous system. And here's a slide from the classic paper looking at basically the mind of a worm and actually mapping this out in the location and within the nematode of all of the different neurons as well as how they're connected. So if you look at this wiring diagram, there are about 300 neurons, and what's neat is that it's invariant between animals. So this is a consistent network. They're also very highly locally connected, and there's about 5,000 chemical synapses or connections. So we can think of these as our edges in our network. And feed-forward loops are found to be highly enriched in this system as well. So again, when the similar sort of approach and statistics are done, so in a defined network in C. elegans, and this one consisted of 252 neurons with 509 connections or edges that they looked at. Within this network, there were 125 feed-forward loops identified within that network, which is enriched relative to what you would expect if the neurons were only randomly connected. So again, we have an enrichment of feed-forward loops in our neuronal network in C. elegans. So for some further reading in this area, I've listed some references, some that we've cited and looked at today that might be really interesting if you're interested in this topic and some want to go a little deeper. And coming up, we'll get into these questions that we've raised about the dynamics of the feed-forward loop, and particularly looking at some of the advanced design features and how we can tune those design features. And these include creating sign-sensitive delays, noise filtering, persistence and coincidence detection, increased response time, and pulse generation, all using different feed-forward loops. So I hope to see you next time.